So you can see my slide?
Yes, ma'am.
Yes, ma'am.
Okay. So what Dr. Sumit asked me to do was just to give a brief overview about hemolytic anemias, and that will be followed by the discussion of the cases, which I'm going to ask the students to kind of chip in and give their opinions. So basically, how do we define hemolytic anemias? It is increased destruction of the red blood cells. And when we get a case of a diagnosis is required for hemolytic anemias, we have to use a stepwise diagnostic algorithm, which starts basically with the clinical examination. So the clinical history is very important, where you will find that most of the patients will present with jaundice. Gallstones are very common finding in patients with chronic hemolytic anemia, and splenomegaly may also be seen. Now, usually most of the times we talk about hemolytic anemias, they're probably acquired conditions are quite common. But in this particular session, we are going to talk about the hereditary causes of hemolytic anemia. So when we talk about the intrinsic disorders of the red blood cells, among the inherited disorders, there are many disorders which are there. They can be classified as three major groups. The first is the globin abnormalities, and we are going to discuss about the globin abnormalities in the next session, next week. Then comes the red cell membrane disorders and the enzymopathy. So red cell membrane disorders are not very common, and so are enzymopathy is not very common. But what is important, I always tell our residents, is that the reticulocyte count is very important, because that is a complete measure of the adequacy of the erythropoietic activity, which we are talking about actually the bone marrow. So the reticulocyte count, many people describe it as it's the window to the bone marrow without actually seeing the bone marrow. So that is something which is you need to do it very diligently. So you might get hemolytic anemia where the patient does not have anemia but has hemolysis. So that we will call it as a compensated hemolysis, and there the marrow actually has the capacity to compensate and increase or normalize the hemoglobin. The other situation, which is more common, is probably that the patient will have anemia of varying degrees, and usually the hemolytic anemias which are inherited are episodic kind of conditions which happen. So what about the how to measure it? So most commonly people do a manual reticulocyte percentages. You must see at least 1,000 RBCs which are stained, and of them see what is the reticulocyte count. Now it is also mentioned in the books that the reticulocyte count, you must see at least 100 reticulocytes, which is not easy to do. So usually we look at one field where we have about roughly 100 RBCs, and we count the percentages by seeing 10 such fields. I always like to see the manual, because in the manual I'm able to see the various types or the phases of the reticulocytes. And maybe Ankita, if you can just tell us about the different types of reticulocytes which are there, or anybody can take it up. Any of the students? What are the different types of reticulocytes?
Four stages will be there, ma'am, in reticulocyte count.
Yes.
Based on the content of the RNA in reticulocyte, the stages will divide.
So when we say type 1, that is the most mature or the most immature?
Most immature, ma'am. Most immature.
Very good. Yeah. So as it matures, we go up to type 4, and sometimes we see them as a very small, tiny dot, something like this. Hardly 1 or 2 dots are there, and some of them are very full of reticulocytes. These are probably majority of the cells that you're seeing are probably type 2, 3 type in between there, and some of them are really loaded with a lot of blue color, and they are the immature ones. Yeah. So the other way of doing it is the automated kind of method. We can use a flow cytometer or the automated blood cell counters nowadays. All of them give us values of the automated reticulocyte counts. So between the manual, which I like to do, and the automated, in the automated, how do we see the parameters? Anybody can answer? Maybe Ami can take up this question. What are the different types of reticulocytes by the automated blood cell counter? Dhruva, do you want to give it a try? Muniswari?
Yes. They do immature reticulocyte fraction, IRF1 parameter will be there. Towards the most right side, this IRF fraction will be there. And as it comes towards the left side, the mature stages will come.
Okay, so we have these parameters which we are able to see on the automated blood cell counters, which is the low fluorescence reticulocytes, the medium fluorescence reticulocytes, and the high fluorescence reticulocytes. So usually when we talk about the immature reticulocyte fraction, which is given here, IRF, that actually gives the total of the medium and the high fluorescence. So most of the people, majority of the people will have low fluorescence reticulocytes, which is the maximum percentage. And if this increases the immature reticulocyte fraction, then we say that there is the bone marrow activity is kind of increased. Another parameter which is used sometimes, not for hemolytic anemias, but reticulocyte hemoglobin is also seen sometimes with anemia of chronic disorders. There's another term which is used, which is known as the reticulocyte production index. And here we have to correct for the degree of anemia. So how we do it is that either whatever is the reticulocyte count that we get, that has to be multiplied by the hemoglobin that we've got divided by the hemoglobin that is expected for the age at that particular time, right? With that, then you have to correct it, because if a person is normal, the anemia should be corrected by the reticulocyte production index. And the RPI of more than 2.5, this indicates hemolysis. So this is a simple kind of a method which is used for correcting and telling, kind of showing that what is the reticulocyte production index. And we know that the reticulocytes normally take 2 to 3 days in the peripheral blood, but sometimes, they are released prematurely from the bone marrow. And I think in the last talk, a very good description about this was, given by Dr. Sunita. So this is the, diagrammatic representation of the red cell membrane, how it looks like. And we have the vertical interactions and the horizontal interactions. And Samina is there. Samina, can you just describe this, what we are seeing on the slide?
This is a structural representation of the RBC membrane. Here we can see vertical interaction between band 3 and chitin, and the horizontal interaction is given by the spectrum, alpha spectrum and beta spectrum.
Okay, so the band 3 is produced as a dimer all the time. So you can see that this dimer is over here, but it is not really forming a pore. So when 2 dimers bind together, so which is a diagrammatic representation here of band 3, so it forms a pore in the red cell membrane. And band 3 will then go and attach to the 4.2 and then the ankylin. The ankylin, a part of it will go and attach to beta spectrin, and beta spectrin will attach to the alpha spectrin. So you have the head-on attachments of the beta spectrin and the alpha spectrin, and that forms that mesh all around. That forms the cytoskeleton by the horizontal interaction. So all these proteins are important. Band 3, protein 4.2, ankylin, beta spectrin, and alpha spectrin. These are the major membrane proteins which are important for us. And you see that multiple, these green colored structures which are there at the top. These are various kinds of membrane-bound antibodies which are formed, and multiple things are there on the membrane. So the red cell membrane is a very, very active kind of a membrane which is there. And this next slide actually is just giving a representation of which are the disorders which are associated with which kind of diseases. So the topping the list here is the hereditary spherocytosis. You can see that the band 3 is also attached to CD47, 4.2 here, and the enchirin. And any interaction here is going to give rise to hereditary spherocytosis. Usually you get hereditary elliptocytosis and hereditary pyropoikilocytosis if there's a problem with the beta and the alpha spectrums. And the other newer kind of membrane proteins which are there are the PISO1 and the KCNN4. These give rise to any abnormality, gives rise to dehydrated hereditary stomatocytosis, and we'll come to it a little later. Right? If we talk about the hereditary enzyme disorders, these are also known as non-spherocytic hemolytic anemias. So when we talk about the membrane disorders, we call it as a spherocytic hemolytic anemia, but the hereditary enzyme disorders, these are known as non-spherocytic hemolytic anemias. Among them, these are the various disorders which are there. Here again, you can see that though the RBC is actually majorly comprised of the hemoglobin molecule which is there, but these enzymes are extremely important to maintain its life cycle. It doesn't have the nucleus, so you know that the RBCs are the platelets. These two are the only cells of the body who don't have the nucleus, but they are very, very active cells, and that is because of so many enzymes which are there, which are very, very important. Of topping the list as the commonest disorder because of enzymopathies is the G6PD deficiency. The second most common disorder is pyruvate kinase deficiency, and pyruvate kinase deficiency is supposed to be the second commonest, but you'll find that in the literature or otherwise it's very not easy to diagnose this because the tests are very difficult to conduct. So I have not put a case of pyruvate kinase deficiency, so I'll just briefly tell here that pyruvate kinase activity in the WBCs is very, very high. So whenever we are doing the assay, we have to remove the WBCs, and that is the tough part, that you have to deplete the blood with WBCs before you can do the test, otherwise you'll get a false negative. And most of the hereditary enzyme disorders have this problem that the enzyme assay is not easy to standardize because you need a fresh blood sample, one. Secondly, because it's an enzyme activity that you are seeing, so all the reagents have to be freshly prepared, and most of the tests you have to do at 37 degrees centigrade. So because of these problems which are there, many times it's not easy to identify these particular conditions. So very uncommonly we do see hexokinase deficiency, the glucose phosphate isomerase deficiency, and among the disorders of the erythrocyte nucleotide metabolism, the commonest is supposed to be the pyrimidine 5' nucleotidase deficiency. I myself have not seen a case till now. I've just seen some case reports, and we know that we get a lot of basophilic stippling in the peripheral blood before when we think of pyrimidine 5' nucleotidase deficiency. I'll ask the chairpersons if they want to chip in here before we go to the cases right now.
I think there is the background and also the brief.
Tufan, you have to be louder. Tufan, we can't hear you.
Yes, I think. Thank you, Renadi.
Your voice is, Tufan,
Still not there.
means it is very low as if you're speaking from another world.
Now it is audible.
I think you're coming close to the earth.
I'm in the close to that. I think.
Now it's better. Now you're near Bombay.
Thank you, Renadi, for this wonderful lecture. And I think the brief discussion is your membrane disorders. And also, of course, the hemolytic anemia you rightly pointed out, because the hemoglobin indices and the reticulocyte count has to be very important for evaluating the any cause of the anemia. And also the beautiful description of this non-spherocytic hemolytic anemia and their tips also. Thank you, Nadi.
So there's a question: do WBCs interfere? What was it? Prashant, can you help with the question? There was a question I said.
Question is, do WBCs interfere with all enzymopathies?
No, it doesn't interfere with all enzymopathies. It's very specific for the pyruvate kinase, because the activity of pyruvate kinase, I don't remember how much, but it's more than 100 times, I think. Though, if you consider the number of WBCs, they're much, much less compared to the RBCs. But the activity is so high that it can overcompensate. So that is the problem. We need to separate out. And there are methods to separate out the WBCs, but it's a little cumbersome in the lab to do it. So we have standardized it, but you can do it only in specialized labs, these kind of assays. It's not very easy to standardize in most of the labs. So we come to the first case. Yes.
Dr. Budeli, you have any comments?
Yes. Madam, sir, can you hear me, sir?
Yes, of course. Yes.
Ma'am, it was a very good start, madam. Brief introduction. And very rarely we come across these kind of cases, only that we read in the textbooks. So it's really informative and very nice beginning, madam. Very interesting cases. Thank you, madam.
So I'll ask Ankita to discuss the first case, maybe just the beginning.
Yes, ma'am. A 42-year male presented with recurrent jaundice for the last 2 decades. On investigation, he was found to have mild anemia, and on examination, moderate splenomegaly was noted. The family history showed his 14-year son had 1 episode of jaundice.
So with this history, what do you think is the pattern of inheritance which is there in this family?
Ma'am, hello?
Yes, we can hear you.
Autosomal dominant.
Very good. So, if you just take a very good history, you can make out that this is an autosomal dominant kind of inheritance pattern which is there. The father has got a problem and the son has also inherited the same problem. So if it was autosomal recessive, what would you have expected in this family?
Ma'am, might be the son must be a carrier and daughter must be having.
Okay, I think you're on the right track, meaning that if we're talking about the index case as the 42 years male, then it is all the children will have autosomal recessive, like at least one carrier kind of a situation. But usually what happens is that autosomal recessive inheritance patterns give rise to disorders very early in the children. So you will find in autosomal recessive conditions, the patient itself would have presented very early and the parents will be asymptomatic, right? So when you look at a family and you find that the presentation is early, then we usually think of autosomal recessive, especially when we look at the parents and find that the parents are completely asymptomatic. So here it is looking like an autosomal dominant kind of condition, which is kind of more common.
Ma'am, can I ask a question?
Yes.
Yes. Why would it not be an X-linked inheritance, like an X-linked father to son? There's only one X chromosome, so if that is a problem in the father and the son inherits that.
So it's a very good question, I will say. The only thing is that, most of the red cell disorders, they don't have an excellent inheritance pattern. So when we talk about X, so if you look at the X chromosome itself, most of the, apart from the G6PD deficiency, which is on the X chromosome, apart from that, the other hemolytic anemias are not there on the X chromosome. So they are there as autosomal genes or that kind of thing. So usually you're right that if with a history like this, the X-linked condition, we'll also not think in that sense because X-linked, the inheritance will come from the mother.
The mother, correct? Yeah. Yes, ma'am.
So the mother would be a carrier and the son would be affected in X-linked disorders. So that is usually happens with hemophilia A, hemophilia B kind of conditions, even with G6PD deficiency. But here it is kind of little different. That's why we don't think about the X-linked conditions. Right?
Yes, the mother would have been involved. Yes.
Mother
Would have been a carrier.
would have been a carrier.
Yes, ma'am. Yes.
She would have been asymptomatic kind of. So when we think about X-linked disorders, there usually you will find that sometimes the cousins will be affected or the mamaji will be affected.
Yes, ma'am.
Maternal uncle basically will be affected. Right?
Yes.
So maybe Dhruva, you can describe the red cell indices here of the index patient and the son.
Yes, ma'am. Ma'am, the hemoglobin is lower in the son. It's low in both the father and the son, of course, 10.2 versus 8.1, but lower in the son. The pack cell, the same with the PCV also. The pack cell volume is lower in the son than the father. RBC count also the same thing. MCV is a little reduced, a little bit in the microcytic category. The MCH is slightly reduced, but not of much. I would not really look at that. MCHC is in the normal range, so this is not a hereditary spherocytosis, or there's no spherocyte. Within this case, there's no spherocyte formation. The RDW is very active, so that would mean that there's some kind of compensation that is occurring from the bone marrow, and there are probably immature cells that are being produced. And yes, there is a large size difference between the cells because of that. Older cells are probably smaller, and the newly produced cells are larger. Reticulocyte is again a giveaway that the bone marrow is trying to compensate, so it's very high in both the father and the son, higher in the son. Lastly, the indirect and direct bilirubin levels, they are now the thing is that if it is a hemolytic process that we are looking at, it would come within the compensated hemolytic category, meaning that it is not so high the level of hemolysis that it is not getting compensated. This is how I would interpret.
But can you tell about the anemia which is there? Do you think it's mild anemia, moderate anemia in the father and the son?
Madam, I think it's moderate sort of anemia in the father. Mild to moderate, I would say. And son, it is more severe.
The father is, yes. So the father is the index case here because he has come with a major presentation. So the son is just getting identified early because the father is symptomatic kind of. Now, Dhruva said about that MCV, MCH being low specifically for a hereditary spherocytosis. Would you agree with that?
Which one, madam? Sorry, MCHC.
MCH. MCV and MCH are mildly reduced.
Yes, ma'am. Because it's a spherocyte, the MCV is expected to be reduced in hereditary spherocytosis. But as he said, the MCHC is maintained in the normal range, even though we have learned it could be increased, considering a spherocyte is more dense and more compact.
So can you describe the peripheral smear, the reticulocyte preparation?
Yes, ma'am. So you can see that there is anisopoikilocytosis, the peripheral smear with spherocytes of varying size and shape. We can see some few schistocytes.
So will we call it schistocyte because the cell which is here, it's looking a bit dense and then it has some spiny processes. So will we call this a schistocyte or some other type of cell? Anybody else would want to give it?
Acanthocytes.
Acanthocytes will be fine. So we are seeing quite a few spherocytes here, and also some of them are normocytic. But you can see that the ones which we are calling spherocytes, they are quite dense. The hemoglobin is quite dense. The central pallid is lacking, and the size is looking to be smaller. And in addition, we have some acanthocytes. So do you think this acanthocyte should be there in spherocytic anemias?
Ma'am, it would be artifactual, ma'am.
Artifactual also. Yeah, somebody was saying.
Ma'am, in case of hemolysis, it can be normally seen in the smears. That's what I think of. The sign of hemolysis.
Hemolytic anemia. So in this smear, I think one description which was missing was that RBC density is looking to be reduced also. In the smear, at least what is there. If the smear has been made well. And we don't see that much of polychromasia, but usually see polychromatophils also. Maybe a few of them are polychromatophils, but not as much as we would have expected probably. What about the reticulocyte preparation?
Ma'am, it's increasing reticulocyte count is there.
Yeah. So usually when we look at the reticulocytes smear, it is a bit patchy sometimes. And reticulocytes, because of the negative charge which is there on the surface, they have a tendency to aggregate. So you need to see when the reticulocyte count is more, then you need to see more number of fields. Usually you'll see so many, you'll find the thing that, okay, let me see only a few fields. But it's a good idea to see multiple fields and then make up your mind that what is the percentage. So here we can see that there are various stages of the reticulocytes. Some of them are quite blue in color, and others are various stages, immature, and many of them are of various stages that we can see, right? So finally, now you have the whole case in front of you. You have the red cell indices as well as the peripheral smear findings. So what is your opinion, Muniswari?
Ma'am, going according to the peripheral smear, I thought first it's hereditary spherocytosis because I'm seeing some spherocytes. So, but as Drew said, MCHC is not raised, which is going against the hereditary spherocytosis. So, according to the history, yeah, there is autosomal dominant inheritance is there, and patient is also having from past 2 decades is having recurrent types of jaundice and splenomegaly. So all these findings are again going favoring of hereditary spherocytosis. But one finding I'm not able to correlate is at MCHC. So where I got stuck actually.
You got stuck. Okay. So if we think about the causes of spherocytosis, I just want to mention that spherocytosis is a very, very non-specific finding. In multiple conditions, you will find spherocytes, but in this particular case, the major cells which are there are spherocytes. So we try to explain the MCV being less and MCH being less, and MCHC being in the normal range. But you can see that there are various conditions which come to mind when you look at the causes of spherocytosis. Topping the list, of course, will be the transfusions, post-transfusion. That also we see quite commonly, after transfusion. Sometimes Whereas in an iron deficiency anemia case, you will find that they've given blood, and you can see dimorphic populations very nicely, hypochromic microcytic and some spherocytes and some kind of normal kind of RBCs. So it's a good idea to have a look at this. And of course, among the acquired, it is a warm autoimmune hemolytic anemia, which is the commonest cause. Where sometimes, if it is cold type, you can see some clumps also of the RBCs. So any additional history or test that you would like to order? How would you approach a case like this?
First of all, we will go for serum LDH because it will define the severity of hemolysis, and then we will go for serum haptoglobins, which is decreased in case of hemolytic anemia.
Anything else you want to do?
Then we further go for osmotic fragility test. We will prefer incubated osmotic fragility test because it can identify mild spherocytosis also. So
Extremely good. That is a very good approach. So incubated osmotic fragility test is a good test because it gives you a nice graph. And now we know that when we are doing the test, if you have a positive family history, testing with the family members is a good idea. Because if you would have done the mother and the father in addition, you'll find that the mother will be normal, the father is showing, and you know then the pattern of kind of inheritance which is there, if possible to do. Now, what kind of sample should be collected? We should collect the sample in which file?
EDTA.
Yeah, we can use EDTA, but regularly, if you look at DAISY, it mentions that you should use defibrinated blood. And in the lab, we usually collect the blood in heparin. So you can use any of the anticoagulants. So just a very simple question to Smina, that the heparin vial, what color vacutainer is it?
Green.
Very good. So it's a green top vacutainer, the heparin one, and that is kind of preferred nowadays. But in DAISY, it is actually given us So among all the tests which are there, this is the most sensitive test. But the important thing is that you have to collect the blood sample under sterile conditions because you're doing the test after 37 at 37 degrees. You're keeping it overnight. But the next morning you'll be doing the sample. So every time we are doing the test, we must have a control also. So the control is important. And next day we look at the blood sample and see that there's no extra hemolysis. That means that the sample is kept well, and then we can do the incubated osmotic fragility. I think it was very nicely explained in the previous class. The other test, which is nowadays very preferred kind of wherever flow cytometry is available, is known as the eosin 5-maleimide binding test. And I think, Muniswari, you all are doing this test in your department, I presume.
Yes, ma'am. Yes, ma'am. When there is
So-
HS case, we will do this EMA binding test.
So would you like to describe this slide?
Yes, ma'am. So eosin 5-maleimide is a dye, which usually this dye binds to band 3 on the RBC membrane. So if band 3 is there, this will bind this dye will go and bind to band 3, and it will emit the fluorescence when you see in the flow cytometry, and that fluorescence will measure. But in case of hereditary spherocytosis, where there is band 3 deficiency there is like absence, congenital absence of this band 3. So this dye cannot bind to this membrane of the red cell. So the fluorescence will be less when compared to the normal control. So that's how the fluorescence will be little less.
So whenever we are doing this test, one precaution is that at least 5 to 6 normal control, which should be age matched, should be taken. And usually we look at the red cell parameters, that all the parameters are fine. There's no reticulocytosis that will be taken as control. And then we compare it with the patient divided by the mean fluorescence of the normal controls and get the value. So what you mentioned about the band 3 being absent or less is not really true, because we talked about the membrane where we talked about band 3 as the major protein. But apart from that, we have any of the other proteins, that means ankyrin, the 4.2, the alpha spectrin, beta spectrin, any of these could be involved. So this is what I really find this test to be very interesting, because it is always binding at one particular location of the band 3, but it is giving you idea about all the cases of HS. So extremely sensitive test. And in our experience, we have found that even patients who have received, had a splenectomy, so their hemoglobin has normalized. But still, this test, if you do it, you will find that the EMA binding test will always be reduced, kind of. So it's a very, very specific test in that sense. A sensitive test rather. Specific, not so much, because CDA type 2 can also be showing you a reduced level. So,
Yes,
but clinically, you will know that there you have, even if you have anemia, the reticulocyte response is not there in congenital dyserythropoietic anemias. So the differential diagnosis will go from there, and you would have seen spherocytes in the case of HS. So this is quite a good test to be done. So this is just showing that in a patient, the value that we have got is about 2,890 divided by the mean value which is there of 40, which we have got. So the value is less than 0.71. So if it is less than 0.8, if the MCF is less than 0.8, then it is like a HS, but in an appropriate clinical setting. So appropriate meaning that there should be reticulocytosis, clinically jaundice, and those kind of things. History is very important whenever we are interpreting the test, right? So this is mainly about. So we had done this analysis some time back. It was an MD thesis, and we found that this test is a very good test for hereditary spherocytosis. There's another test. If you have a flow cytometry with you, you can also do this test. And can anybody describe this test? Ami, you want to give it a try? Has anybody seen this test being done in the lab? Okay, so this is a very simple test, meaning that you take the blood, and in that, in the normal saline, basically, you're putting, not normal saline, in distilled water, we are putting blood. So when we put blood and we see in a little while, we take the reading, we find that if it is a hemolytic anemia, you'll find that at the end of the reading, which is like a time slot of, I think 1 or 2 minutes only, I've forgotten exactly how much it is, you'll find that the residual cells which are there are very less. So you can see the number of dots which are there in a patient with hemolytic anemia, it is really reduced when you compare it with the normal. So here it's like the amount of RBCs which can sustain the osmotic shock is much, in a normal, the number of RBCs will be much more compared to a case of hemolytic anemia. So this is a good test, in a simple test which can be done if you have a flow cytometry available with you, right? So basically, we are talking about hereditary spherocytosis, and these are the clinical pictures. Hepatosplenomegaly is there, jaundice is there, presence of spherocytes. You may get a positive family history. When you get a positive family history like that, it's easy to diagnose it as a dominant. But we also know that it can be autosomal recessive, and when it is autosomal recessive, like I told, the patient will present early. The parents may not be symptomatic, and so it would be autosomal dominant in almost 80% of the cases. The cases with beta spectrin, alpha spectrin mutations, they usually present as autosomal recessive inheritance, right? So last talk which was given by Dr. Sunita, she also mentioned about this red cell membrane electrophoresis which can be done. And we have tried to do it. Red cell membrane electrophoresis is very difficult to do because we know that the RBCs predominantly contain the hemoglobin, which gives the weight to the RBCs. And the red cell membrane is just 1% of the weight, right? So when we are trying to extract the red cell membrane, it is a difficult task. We have to lyse the RBCs and do a very high-speed centrifugation, multiple centrifugations, so that what is left behind is just the red cell membrane. Then the protein quantitation has to be done. And then we do a electrophoresis, which is a gradient electrophoresis. So the gel, basically, it's a polyacrylamide, SDS polyacrylamide gel, which is a gradient gel. And we look at the bands here. So you can see that if you look at this lane, you can see the upper band, which is the alpha spectrin. The next band is the beta spectrin. Now, ankylin is one of the proteins which has multiple isoforms. So I don't want to get into the complications because it's not easy to remember so much, but it's very difficult to make out, that which is normal, which is not normal, kind of. So this test, I would say, is kind of abandoned. It looks interesting. We talk about it, but it's not a very easy test to standardize in the lab, and it's not really required to be done in a routine kind of lab, right? So in the pathogenesis of any of the deficiencies which are there, meaning whichever protein is deficient, the end effect is that your band 3 is reduced, and that's why your EMA dye test is going to be reduced in the flow cytometry. So in our study, we found that this MCHC that we give so much importance to as being more than 35.9 gram percent, we found that only 25% of our hereditary spherocytosis cases had this. We don't know the reason for that. Maybe there is iron deficiency anemia, which is still there in these patients, but it is supposed to be a sensitive kind of a test. But in our experience, we have not found it to be that sensitive. And just as a take-home message is that molecular diagnosis is not required for hereditary spherocytosis. If you have the clinical spectrum, it's more than enough. Nowadays, because the next generation sequencing and molecular diagnostics have become more easily available, I will say. So because of that, people before doing the clinicians are asking for this molecular diagnosis because before doing a splenectomy. But otherwise, for a diagnosis, only a clinical picture, presence of spherocytes, episodic kind of history, and the presence of incubated osmotic fragility is enough for you to make a diagnosis. I hope this is clear, and if the chairpersons want to add anything, I'll be happy.
I think there's a beautiful case has been described by Dr. Ina Das. I wanted to comment on this issue in terms of anemia degree. That is for the student only. The mild, moderate, and the severe. If it is less than 7 gram, we call the severe. In 7 to 10, we call the moderate, and 7 to 12, we call the mild degree of the anemia. Another thing is that in the cases, there is a microcytic hypochromic and the RDW is slightly raised. So along with this osmotic fragility, as Dr. Ina Das rightly pointed out, so I'll also doing some iron study to rule out the co-existing iron deficiency anemia also. And another important thing is that the transfusion thing is very, very important because when you're doing the osmotic fragility test, so not to take the transfusion history of the patient. Because if the patient has been recently transfused and you have been asked to do the osmotic fragility, then your test will be the fallacious. So I think these 2 to 3 things is very, very important. And this thing has been beautifully described by Professor Das. Over to Dr. Das.
So we'll go to the next case, and I can ask Aishwarya to present this case.
Ma'am, before that, there was another question regarding EMA binding. So can you take that up? Yes, ma'am. So if Jamal asks, just want to confirm, I think, so EMA binding isn't specific for band 3 only. It can be decreased in band 4.2 and can inspect in deficiency cases also.
Yes, any of the cases of hereditary spherocytosis will show a reduction in the EMA binding test. Yeah, that is the beauty of the test. That though we are, studying only the band 3, and that kind of gives us an insight that how beautiful the interaction is there in the nature. That when you look at the diagram, also you see that they are really just touching each other in the interaction, but it's a strong interaction wherever they are binding to each other. So that the end result is that the band 3 is looking to be deficient when we are studying the case.
Right. So I think in the Southeast Asian monocytosis and the xerocytosis, these are the 2 conditions. The EMA test might be problematic. It sometimes gives the negative result also.
So Southeast ovalostosis we really don't get here. That is the we haven't yet seen any case till now.
Right.
So CD type 2 is the other one which can give rise to the overlap with the EMA test. Yeah.
Right.
Go on to case 2 now.
Yeah. Can we go to the case 2?
Yes, so the case 2 is of a 9-year-old male who presented with fever and clinical features of renal failure. So the values, hemoglobin is markedly reduced, severe anemia. RBC count is reduced. The MCV is in the higher side, it is 105. MCH is on the higher side, and MCHC is normal. The red cell distribution width is increased, and there is severe reticulocytosis, that is 21. The total count is normal, and the platelet is also in the normal range.
So this is the peripheral smear.
So there is abundant microspherocytes. Then you can see a few. I'm thinking it's polychromatic cells. And then I can see I don't know, suspicious bite cell in the center. I don't know if it's a bite cell. Then there is an nucleated RBC.
Very good. There's a nucleated RBC also. So if you look at the smear, actually you tend to feel that there are more spherocytes than anything. But if you compare it with the previous slide, you can see that spherocytes are there, but there's some membrane irregularity, some little snouts are there. And if you look closely, maybe we can see that the hemoglobin has contracted on one side.
Blister RBCs.
Okay. So some of them are like blisters here, you can see, no? So it's looking quite abnormal. So so what would be your possibility?
Ma'am, are we seeing helmet cells, degmacytes? I feel I'm seeing a lot of helmet cells here also, like, and more schistocytes also.
Histiocytes also I'm seeing.
And is there a dual population, like some large cells and like some
Yes.
macrocytes and some schistocytes and smaller cells?
Yes. So the macrocytes that we are seeing, many of them are probably a bit polychromatic. Like the staining is not excellent, but there's fewer cells are large actually. But a majority of the cells are looking spherocytic. But they have if you look closely, actually there's a membrane which has got retracted. So what is looking like a helmet, if you look closely, actually the membrane is still intact. Some of them, of course, it is pinched off. And do we know the reason why it is getting pinched off? The RBC membrane.
RBC, that hemoglobin is contract with due to that, and that's splenic macrophages, so then that removes the hindbody, due to which that form a blister shape structure.
Excellent. So we are getting many of these cells, and so if you can make up the clinical picture, then can you suggest some diagnosis in this, or what test should we do?
It's showing acute hemolytic anemia on the basis of clinical history, peripheral examination, and investigation. It is showing the case of acute hemolytic anemia.
What are the possibilities that we can think about seeing the cells and everything?
And we will take the further history because on clinical findings, only 2 histories are given, like fever and clinical features of renal failure. So we will take the history of whether there is any exposure to drugs or like anti-malarial or sulfur drugs like Dapsone or Septran, or whether in the past history he had an infection like free oxygen derived free radical injury, or like any exposure to foods like flour beans.
So what are you pointing at because of all this history that you're keeping?
Ma'am, as the peripheral smear is typically showing that white cells and blister cells, they are pathognomic of G6PD deficiency. So we will go for the history first.
History, and then you can also do look for these Heinz bodies which may be there. Heinz bodies we can study because we know that G6PD variants can have these various different classes of different types, which is the commonest one we can see is class 3. This is the moderate enzyme deficiency where you get intermittent hemolysis, and there's an acute exacerbation because of either infection or drugs kind of. So this is the more common one, but very severe deficiency can give rise to chronic hemolytic anemia of the non-spherocytic type. Usually the children will present very early in age. So this is not a very common condition, the class 1. The class 3 is much more commoner, and with the type of G6PD deficiency which is more commonly seen in India.
Mediterranean type.
Very good. Very good. G6PD Mediterranean, that is the more common one. And we talk about this G6PD being common in our population because of malaria being prevalent. Endemicity because of the balanced polymorphism that is there. Because the RBCs which are G6PD deficient, they are able to resist malaria basically. So if RBC is infested with a malarial parasite and it's G6PD deficient also, so that parasite will the RBC will get lysed. So that's not very good for the malarial parasite. So we feel that probably the frequency of G6PD deficiency is higher because of malaria being there in the tropical countries. So that is one of the reasons they say which is there, right? So this is just giving the pathogenesis of the hemolysis which is there. So basically there's an oxidant stress because of that. You get the production of denatured hemoglobin, denatured globin or sulfhemoglobin which binds to the surface membrane, and it gives rise to production of Heinz bodies, and then you get both extravascular as well as intravascular hemolysis, right? And these are the various tests. I think Dr. Sunita had very nicely described these tests also, that this is the common test which is done. This is not a quantitative one but a qualitative test, and basically we put blood and sodium nitrite with methylene blue. The methylene blue helps in converting it back to from oxyhemoglobin. Basically the conversion is very good, but if there is deficiency, this conversion from methemoglobin back to oxy does not happen. So this is one of the tests which is done, but just a point to remember that for this test you need a time period of about at least 3 hours to do the test. So if somebody comes at later in the day, then it's not easy to do this test. But this is, since it's a very simple test, it's not expensive, and the problem is that this is not a quantitative test. So for screening this is a good test, but if you want to do a quantitation of the enzyme, then there are other methods which are there. In the field, many people want to do this fluorescence spot test because you can make the blots and you can transport the filter papers and do the test later on in the lab. So this is a fluorescence spot test is also a good test if you're trying to identify a female with G6PD deficiency, because the carriers will give you indeterminate kind of results basically. So the spot 8 which is there, this is a hemizygous kind of heterozygous female, and for the males who are deficient we say hemizygous males because the males have only one X chromosome, so they will be symptomatic with G6PD deficiency, and the mothers will be carriers. So this is the X-linked recessive disorder that we say. So the males, hemizygous males are going to be affected, and the heterozygous females are usually not affected, but you can get a situation where the female has got the both X chromosomes, one from the mother and one from the father. Both of them are deficient. If both are deficient, then she will become a homozygous female. Not a very common condition, but it is possible to get like that. The females can also be G6PD deficient, but either because of this Lyne's hypothesis, which is like the inactivation is the normal is inactivated more than the G6PD deficient one. So we can just remember in general that this is an excellent recessive condition, and usually the mothers will not be symptomatic, right? So we can go quickly to the case 3, which can I think Amy can describe this.
26-year-old male female with the feature of the chronic hemolysis was on the intermittent follow-up. Clinical and laboratory finding included short unconjugated hyperbilirubinemia, hepatosplenomegaly, polylysis. Family history was negative. So, ma'am, it's not an inheritance disorder, and in peripheral smear, there are the somatocytes seen, and there is the presence of the large platelet also, ma'am, inside the
Polychromatics also.
Very, very. So we have got polychromatic. So here you can see that almost 80-85% of the RBCs are looking like stomatocytes, which are there, right? And like I said, some spherocytes are also there, and some of the RBCs don't have that central pallor kind of, and then you have some giant platelets which are there, and polychromasia is quite marked, right? So what do you think is the possibility? I think Pandika is there.
Ma'am, maybe it's the Mediterranean stomatocytosis.
Very good. That's a very, very good possibility that we think of. What do you think is the pathogenesis of Mediterranean stomatocytosis? So this particular case, she also had a brother. So both of them were symptomatic. You can see that multiple, stomatocytes are there in both. The numbers are variable. The reticulocyte also is increased, but the anemia was not very severe. We also saw these giant platelets which were there. So usually what happens is that if this condition is not seen or identified properly, the thrombocytopenia becomes something which is very important. And in this particular case, actually a bone marrow had also been done, and we had called it as a megakaryocytic thrombocytopenia. This is one of the very old cases that we had. At that time, we didn't know about this condition being there as Mediterranean stomatocytosis. And because it is compensated hemolysis in many of the cases, they are not identified very easily, right? But you have very correctly identified it and talked about it. This is a description of stomatocytic hemolysis with macrothrombocytopenia, also known as Mediterranean stomatocytosis. So the genes which are implicated are ABCG5 and ABCG8. These are basically there on chromosome 2, but we don't need to remember so much of these. Usually, this is an autosomal recessive condition. So if we test the parents, we'll find that some of them will have some stomatocytes which are there, and people are now identifying this condition actively. Earlier, we didn't know very much about this Mediterranean stomatocytosis, but if you look at a smear, it is very, very apparent that there are so many stomatocytes, right? So this is a nice case, and this is just a table to show the various stomatocytic disorders which are there. So this Mediterranean stomatocytosis is also known as cytosterolemia. So basically, what happens is that there is the mutation gives rise to this protein sterolin which is defective, and the plant sterols which are transported. Basically, Munishwari, I think you know about this condition. Can you talk about it?
effect of this ACG5 and G2, there is more absorption of these plant-based sterols from the gut. So there is increased absorption of these plant-based sterols, and these patients they present with mostly hypercholesterolemia. So initially they will diagnose as familial hypercholesterolemia because clinically also they present as xanthomas clinical manifestations. So they'll be initially misdiagnosed as familial hypercholesterolemia because even the laboratory testing of these plant-based sterols, they're mimics like cholesterol only. So it's one of the differential is the familial hypercholesterolemia. So with this when you have this hypercholesterolemia, but the difference from this familial hypercholesterolemia is the presence of these chain platelets along with the stomatocytes on the peripheral smear. When comparing this peripheral smear, we can rule out the familial hypercholesterolemia condition. So this much I know about this condition.
Right. So you can see that there are other stomatocytic disorders which are there. Hereditary stomatocytosis, which can
yes, ma'am. There is two types of overhydrated stomatocytosis and dehydrated stomatocytosis. There's two conditions is there where this overhydrated, both of them will differ with MCHC. I think I'm
yes, you're right.
MCV
MCV and MCHC both are increased in over that dehydrated condition, and in overhydrated MCHC is decreased.
Yeah. Okay. So basically, these are not very common conditions, but hereditary xerocytosis is not uncommon. It is not picked up very easily, but if you look at the MCHC, you can suspect actually. So the red cell indices are extremely important in any of the hemolytic anemias to have a good look at it, right? Okay. Then we come to the case 4, which can be described by Dhruva. I don't see Zahir. Is Zahir around? Then maybe Zahir can contribute. I think he had difficulty in connection, maybe. Okay, Dhruva, I think maybe you can take it up.
Yes, ma'am. 6-month-old child, female child, presented with pallor and mild jaundice for 2 months. Okay, so she was born to a gravida 2 mother from a non-consanguineous marriage. History of severe neonatal hyperbilirubinemia on day 1 of life. Bilirubin 38 milligram per deciliter. Double volume exchange transfusion was performed 3 times over 1 week. So I feel that this was some, like on the first day there was severe neonatal hyperbilirubinemia due to some kind of a hemolytic process which started in utero. So like in the first week of life, newborn, the RBC membranes are delicate, and the UTP, leukotrienol transferase enzyme in the liver is also not functioning well. So there's a larger, at least a greater manifestation of these hyperbilirubinemia related symptoms. So there could be some mild condition that this child had, some hemolytic condition that the child has, like Gilbert's or G6PD deficiency, or something that persisted, of course, but it was more severe in the first week of life. This is what I was thinking.
Can you describe the peripheral smear?
Ma'am, there are a lot of Burr cells or echinocytes, as you would call them. And
we have a couple of spherocytes again, like I said,
spherocytes.
And then some polychromasia as well.
Yes, ma'am. Some polychromasia, some spherocytes, and some Burr cells, and
I think they're
Acanthocytes.
Echinocytes we'll say when they'll be kind of regular. So usually with liver enzyme. Yeah.
Ma'am, there are some cells looking like a prickle cells which are having its pointed ends.
That's right.
Yes. Towards the bottom.
So again, we looked at the reticulocyte.
Reticulocyte count is elevated, so I mean you can see reticulocytes there. So bone marrow not significant.
So what can we do further now?
Now,
most importantly, that it's a children, and that she or he is showing the history of exchange transfusion for female child. So first thing which came in mind is ABO blood group incompatibility type. So we will go for ABO blood grouping. Next thing is any kind of enzymopathies or any enzyme defects or spherocytic hemolytic anemia type. So we will first go for enzyme test and.
Right. So here again we see that the presentation is very, very early. So we are probably looking at an autosomal recessive condition. Very early presentation. And among these three, the pyruvate kinase deficiency, the hexokinase deficiency, the glucose phosphate isomerase deficiency, these are the common ones who present very early. The hexokinase also, pyruvate kinase actually has a varied presentation. You can have very early presentation. You can also have a later presentation. So there's a lot of heterogeneity clinically. So you can see that because there are so many conditions which are there, nowadays it's become very tough to standardize each and every type of enzymopathy diagnosis. And because of that, nowadays we are going mainly for the inheritance pattern straight away. If you do NGS, where next generation sequencing will be able to give us hopefully in some of the cases a diagnostic clue. So in this particular case, this was we could make out that this is a GPI, the glucose phosphate isomerase deficiency. And here you can see that the patient is actually, it should have been a G here, which means that it should have been black here, but here it is a green. So this is by sequencing, we have kind of confirmed that this is like a homozygous mutation. So in cases like this, I wouldn't like to do so many lab tests because we'll not really come to a definitive diagnosis. So the genetics will really help in. So any undiagnosed hemolytic anemia, probably we should go for genetic evaluation. And here you can see that the parents are both heterozygote. So here this is overlapping the green with the black so nicely that it's like you're finding difficult to separate out the two peaks which are there. And in this one, you can see very nicely that because the presentation is so early and it's also severe, prenatal diagnosis can be offered in these very severe conditions of enzymopathies basically. So I was actually keeping a tab at the time also, and because of that, I haven't really stopped in between the cases to talk about the various, ask the chairpersons to really go in. But I'll just wind up by saying that for hemoglobinopathies, we usually do the hemoglobin HPLC and the various tests that we'll be talking about in the next class. And the red cell membrane disorders, we look at the incubated osmotic fragility test. We also do the direct Coombs test to exclude that we're not talking about autoimmune hemolytic anemias. And the flow cytometry by EMA is a good test. And for the enzymopathies, I told you that we can see hemighosts. Like in this one, you can see very nicely the blister cell which is there. So on a lower part, you'll see them looking like spherocytes, but they are actually like the blister cells. And the diagnosis is always a kind of a problem with enzymopathies because when the patient comes to us, the patient is having hemolysis. And because of that, they are basically immature reticulocytes or immature RBCs which are there who have a higher content of the enzymes. So when we do the lab tests, we find that the enzyme levels come to be normal. So that is one of the fallacies of the enzyme disorders that is there. And I think this is my last slide. So I'll hand it over to the moderators and the chairpersons for the comments and any questions which are there.
I have one question.
Ma'am. Ma'am, I have one question.
Yes.
Ma'am, regarding the second case, ma'am, the 9-year-old male child with fever and renal failure, and we saw a few, showed us a blister cell there, and we called it G6PD deficiency. But there were a few schistocytes and few schistocytes there that I could see as well, like cells that were like that. Why couldn't it be a thrombotic microangiopathy-like condition, an HUS-like condition was what I was
Yes.
fit.
Even I thought it's a HUS condition because it's there is fever in the child and renal failure is there, and we are seeing obviously schistocytes are there. Why can't we think as it is a microangiopathy hemolytic anemia, ma'am? That's a possible difference. We can keep it off.
Yeah, but here if you see, they're not really like schistocytes so much. If you look at, like, this is a very extreme case that I'm showing you. You don't see so many cells. Usual cases will not. Here, the child actually presented as a crisis to us. And that's why you are seeing so many of them. But I think HUS is a good possibility. And also, there you will get thrombocytopenia, I think so.
Yeah, yes, ma'am. That's one finding is going against HUS. The platelets are absolutely normal in this one. So which is going against that.
If you look closely, they're not really helmet cells here. So here you can see that the RBCs, these are almost like some parts are getting pinched off. If you see here, look at the membrane. It's like as if the Heinz body is there, which is, being pinched off kind of. So these are very subtle findings. I think we have to keep our minds open because this session was like hereditary kind of enzymopathies and red cell disorders. That's why probably, but I think you're absolutely right when you say that, we need to exclude these conditions. It could be a congenital HUS for all you know. That is also not uncommon. Common, it's rare.
Atypical HUS.
Atypical HUS.
Yes, these are possibilities which we need to keep in mind. You're very right, yeah.
Ma'am, I also have got for the 3 hereditary somatocytosis. Ma'am, in this, it's ma'am, next.
Yes.
Ma'am, इसमें MCV and MCH both are showing. Ma'am, इसका जो platelet histogram है. Ma'am, just because of this giant platelet, we are going for diagnosis of
Mediterranean stomatocytosis.
Yes, ma'am.
Cytosidolemia. No, this is just, when we get a case, you need to look at the entire case holistically. So here you have an autosomal recessive condition where the brother and the sister both are affected, and you have seen giant platelets. So ultimately, the diagnosis actually has come through genetics only. But we can think about the possibilities. So here, if you see a case like this, you will say that this is looking like a stomatocytic disorder. If you look at the clinical picture here, the clinical picture says that there is a short stage. So that tells you that this is like a chronic condition which is happening. This is not something acute which has happened, And if you look at if you read about the cytosidolemia, you'll find that this is one condition, like we say, that there are some conditions which can give rise to a lot of pleiotropic effects, right? So if you read one of the articles which I've given here, this particular article, this is,
Yes, I just read this article.
Read this article. I
Yes,
think they probably talked about some 4-5 cases here. And if you go through the cases, there is a huge variation about the clinical picture. And that is what is so interesting. That sometimes, the patients can present with autoimmune manifestations for the time being. Then the patient has, ANA has come positive, has gone down. Then there is, we've had a family where the girl actually presented as a Turner syndrome. Streak ovaries. So what happens is that this phytosterol, which is there, this gets deposited in various organs of the body. So the xanthoma is just one of the clinical manifestation in some of the cases that you might get. People have seen this to be presenting as young myocardial infarction. So there's a huge, variation in the clinical presentation. Also, the good thing is that there is a drug which is there. Ezetimibe. Yes, that drug is there, which is useful for these patients, and it is FDA approved, not a very expensive drug. The other thing which you can tell the patients is that they should not have oils which are coming from vegetables. Right. So usually we tell them that you can have a lot of ghee. Ghee is a good source of oil, and to refrain from not to have nuts. Not to have sources from where you can get vegetable oils, basically. So that is one of the advice that we can give. So, like I said, this is a condition which is not very commonly diagnosed, and we've just about started, identifying cases because, like, I look at the peripheral smears and very closely for these kind of things, so it's easy for me to pick up. But even for cases where you get, say, I've got lymphoma cases, and I look at the smear, but because I'm looking at red cells, I look at them very, very closely, and I've diagnosed Mediterranean stomatocytosis in patients with leukemias and things like that, erythrocytosis, because my mind is set for the red cells. So you'll have these conditions all over the place, but you just need to be aware of these conditions and look at the red cell morphology. And whenever you look at the RBC morphology, always ask for the reticulocyte count. So that one test which is important is actually the reticulocyte count, which is going to tell you that there is hemolysis going on. So that is extremely important. Yeah, whether compensated, uncompensated, but that is very important as a single test.
Thank you so much, ma'am.
Any comments from the moderator, from the chairpersons? Tufan?
Yes, can you hear me?
We can hear you.
Yes, I think thank you, Rinadi, for this wonderful cases and also the discussion among the postgraduate. There is a good interactions, and I think I will learn a lot from the Rinadi and from the interactions. I think thanks to Dr. Gujral also for arranging such kind of the seminar and inviting me. So thank you very much, and there is no more comment on this issue. Over to Algarani, please.
Dr. Vine is there?
Yes, sir. Yes, sir. Sir, wonderful sessions we have been having here, and a very good set of cases, sir. The spectrum of enzyme defects and also membrane defect, the approach towards it, the pathogenesis, we have learned very well. And Madam has described each and every case, and the approach to every case made the postgraduates understand. Valuable sessions, sir. Thank you for giving me an opportunity to be the chairperson. Thank you, sir. Thank you, sir.
Thank you, madam. Reena, you have any final comments?
No, I don't think so. I've talked too much.
You have my colleagues Aparna and Prashant.
Madam, one doubt, madam. Is it easy to identify spherocytes in a patient who has undergone splenectomy?
Yes, even after splenectomy, the spherocytes, even if the hemoglobin is normal, kind of, you will see a few spherocytes which are there. So after splenectomy, usually you will find that the reticulocyte count still remains high, though the hemoglobin will get compensated. Usually after the splenectomy, the hemoglobin normalizes, but the reticulocytosis will be there. Usually just after splenectomy, you'll find that a lot of thrombocytosis is there for the initial couple of months, but then you might get nucleated RBCs in the peripheral smear. And when you see a situation like that and you still don't know the diagnosis, I think EMA test is a very good test to be done.
Okay, madam. Thank you.
Prashant?
Right. No, sir, no questions from my side. I can just share the talk which is coming up next. And for some students, if they don't, if they haven't joined the Telegram group already, I can share the QR code.
That'll be great.
Yes, sir. So the talk next is on Tuesday, that is 23rd of January by Dr. Ina Das again. And the topic will be overview of disorders of globin synthesis on thalassemias. And Dr. Mamta Manglani will be chairing the session then. And this is the QR code for the Telegram group. So if you are not already a part of this, do scan this code and join the group so that you can receive regular updates regarding our sessions and the links to join the sessions as well.
Thank you, Prashant.
Thank you, sir.
Thank you, Parna and Reena and Tufan and Dr. Vahini and